Hello, and thanks for joining me today. My name is Jeff Prevatt. I am a Deputy Director with Pima County Wastewater, and I'm also have an extensive background in molecular and atomic spectroscopy. For the past 3 years, the United States EPA contracted with the University of Arizona, the Denton Research Group, and myself to help develop standard protocols for the preparation, sampling, and analysis of microplastics. The title of my talk is A Need for Standardized Monitoring Methodologies in Microplastics Analysis, and I hope you'll find some information covered of value, and feel free to reach out to me at the end with any questions you may have. We've all seen photographs similar to the one depicted in this slide, with lots of plastics contamination, plastic bottles, styrofoam, floating on the edges of riverbanks and in the oceans. We face a worldwide challenge and environmental dilemma in regards to plastics recycling and disposal. When we see images like this, we often think of third world or developing countries. However, we see problems like this everywhere. The photo on the left, this is one of the well-known Pacific gyres. It's just a massive heap of floating material, styrofoam, plastics, all that type of material. We typically see pollution like this at the macro level with full-size plastic bottles and just discarded debris. This 2020 New York Times article kind of brings it home a little closer. You'll see a large metropolitan facility in the background with tons of floating debris all along the shores and beach lines. And the article indicates there's nearly 16 million tons below the surface that we don't see. When we discuss microplastics research, we're typically talking fragments of these plastic bottles and waste debris on the order of 1 micron to 5,000 microns, or 5 millimeters. Anything smaller than 1 micron would be considered nanoplastics. Anything larger than that, well, that's macro scale, stuff you can visually see with your eyes. And this 2020 article suggests that the pollution may be far worse than previously thought, even double that. When EPA contracted us for this work, there were 5 goals in mind for ASTM procedure: positive polymer identification, quantification of the number of particles in a sample, the size distribution of particles in a sample, and preferably the shapes, differentiating fibers versus breakdown products of plastics, and preferably a total mass content. The image on the left is from January 2021. And it implicates clothes washing as a major source of microplastics contamination. And when you think about it, it's probably accurate. Consider for a moment the amount of dryer lint that we all pull out of our regular clothes washing dryers. It's usually comprised of human hair, lots of cotton fibers, but also lots of synthetic fibers too. And when you consider the amount of athletic clothing that we all enjoy, whether it's Under Armour gear, dry fit type anti-sweat workout gear, even very common yoga pants, we tend to see lots of these synthetic fibers making its way into our clothing styles in a deviation away from cotton. We tend to notice all that dryer lint because our dryer has a lint screen, but equal amount of those fragments also come out in the wash and are discharged into public sewers. Because there's not a screen on the wash, we don't see it. The image on the left was from an article that came out just 6 months ago in July of 2020, implicating that ocean plastics could triple by 2040. The number of plastics could outnumber fish in the oceans by 2050. This is a very bold statement. Could it be true, and could it ever be verified? These are just some of the environmental implications that we see in headlines worldwide. This 2018 article first chronicled the detection of microplastics in human stools. From a human health perspective, these are just one of many articles depicting the severe health effects. And this article was followed up 1 week later by a more skepticism type article that says there's a lack of analytical methods and valid studies corroborate any kind of health clinical damage from these microplastics. But that doesn't stop sensational headlines. They call microplastics an emerging health crisis. We've all heard of emerging contaminants. Well, they're considering microplastics an emerging health crisis, with many studies indicating that they find plastic particles in bottled beverages, even purified water, processed foods, and many seafoods, with 93% of all bottled water samples containing microplastics. Even more alarming are studies suggesting organ toxicity and impending health crisis, with toxicity to the brain, liver, lungs, and intestinal tracts from ingesting microplastics. This 2019 USA Today article chronicles a study out of the University of Newcastle, Australia, that indicates that the average human on the planet consumes over 2,000 pieces of plastic every week, or 21 grams a month. That's the equivalent of eating 52 credit cards per year. So these headlines seem to indicate that there's an impending public health crisis through the ingestion of these plastic particles. And in this study, October of 2020 indicates they detected high levels of microplastics leaking from baby bottles used for formula preparation. This article chronicles over 16 million microplastics with trillions of even smaller nanoplastics per liter, which makes me wonder who is making these bottles and is there any quality control criteria. Just last month, January 2021, was this article indicating that plastics pollution is showing up everywhere scientists look. Well, if it's showing up everywhere, it's either ubiquitous and we have massive widespread contamination worldwide, or could it be that there's an error in our data and our method of analysis? Those are key questions to be considered. Which brings me back to this 2019 article. This study reviewed over 50 publications dealing with the analysis of microplastics, and they didn't choose complicated matrices like sewage or fish tissues. This was done on freshwater and drinking water samples, some of the cleanest sample matrices one would think they could encounter. In the review of these 50 research articles, the conclusion was contamination is unavoidable. In only 32 of the 50 studies were procedural blanks analyzed, and in only 3 of the 50 samples were procedural spikes used. This lack of data integrity and method validation calls into question some of the results. I remember early on in my graduate career with Bonner Denton, he expressed this phrase often: bad data is worse than no data at all. It impedes your decision-making abilities. It exacerbates the problem you're attempting to study. It can negatively impact your reputation, and it could have severe cost implications. Which brings us to the current state of our research effort, chronicled over the last 3 years. In total, we chose these 13 analytical methods for the basis of our study. One, because being spectroscopists by nature, these FTIR and Raman spectroscopy methods allowed for positive identification against known libraries of compounds. They also allowed for visual identification of particles, which aided in quantification. Now, there are some research groups out there that prefer GC pyrolysis, but that's a destructive technique, which, while it does positively identify the polymer type, it gives you a total mass number. It's a destructive technique, and so you lose out on the finer aspects as far as the number of particles and the size distribution of particles. In particular, consider the cost of these different analytical methods. They range in price wildly from $220 a sample to over $4,000 for a sample. This is due to the amount of time required to prepare the sample for analysis, and also the length of time for analysis on the equipment. If you're using traditional micro FTIR or micro Raman using a rastering type stage, your analysis times could be multiple days. If you're using a focal plane array detector, your analysis times can easily be 24 hours or greater. So starting at the beginning with sample collection, you'll see that most sample collection techniques relied on two methodologies: either sieving of a sample, allowing you to get specific size fractions, or filtering a sample. I prefer sieving. Sieving, you can purchase sieves with different pore diameters, whereas filters tend to often clog and be problematic. And the sieves can be stacked from larger diameter down to smaller diameter, allowing you to collect specific fragments or particle size. And this may be very beneficial to some of these bottled water distributions in determining where in the process are the particles coming. Are they coming from the actual blow injection of the bottles? Are they coming from the screw-on lids? Or is there delamination of the polymeric membranes in the RO systems? There was also considerable variation in the sample size used for each of these studies. Collecting a representative sample is absolutely necessary, especially when trying to extrapolate and quantify for larger operations. For example, if you're trying to characterize the discharge from let's say a 20 million gallon a day wastewater treatment facility and the amount of polymers that are discharged to the environment, you want to capture the entire diurnal pattern throughout the day. So a 24-hour composite is probably the best route. And you'll see that some of these studies used as small as 700 mils to just over a liter. Now here's the problem. If you're using such a small sample volume and you do have natural contamination either in your sample collection or your sample preparation, or your sample preparation is so vigorous that it breaks down particles into multiple particle components, when you try to extrapolate that 1 liter to a 20 million gallon, you're going to vastly overestimate the number of particles. So there's definitely a need for large volumes for representative consistency. Because we are spectroscopists, we chose to stick with the analytical methods, typically involved around micro FTIR, micro Raman, but there was one that looked at stereoscopic visual identification. These visual methods of identification are grossly inaccurate. They're unable to distinguish one plastic polymer from another. We've even seen some where they use a hot pin to touch the plastic particle and see if it melts. If it does, well, then it's confirmed plastics. But really that has all the scientific integrity of a coin toss and should be discarded. We need these methods that we can compare the plastics against a known library. And this is where GC mass spectroscopy works as well, but then you lose the value of sample particle size and number distribution. So the following slides depict the questions that EPA posed of us in developing this method. Our goal was to add greater analytical robustness and greater data integrity. So in order for a method to be robust, we need standardization and reproducibility. And that reproducibility has to occur at the sample collection, sample preparation, and throughout sample analysis. I'm assuming most of the people listening are laboratory personnel, and therefore you're very accustomed to using SOP, standard operating procedures, for sample collection, preparation, and analysis. And that's what we're trying to establish here. Reproducible methods that would tender the same type of results regardless of which lab or who was doing the preparation and analysis. So the primary goal for sample collection is one, that it's straightforward, it's simple to follow, and it's reproducive as possible. These were the goals for any analytical sampling methodology. And because we were using wastewater matrices, EPA selected us because we're a wastewater utility and they wanted to start with one of the more difficult sample preparation and difficult matrices available. If it would work in wastewater, then it was undoubtedly going to work in surface waters and drinking water samples. And so the cartoon on the left depicts some of the necessary components you'll need to do this. Because most of the tankage in a wastewater treatment plant are below grade, you're going to need a pump capable of pumping with considerable head, 10 to 20 feet. The flow rate we selected was 3 gallons per minute. That was selected so as not to overwhelm the sieve stack. Now, if you're doing influent, you're probably going to have a lot of debris and cellulose material, lots of toilet paper in influent, and they'll tend to clog up the sieve stack. And so we do a stacked set of sieves that we can pull out particular fractions, 300 micron fraction, 100 micron fractions. If you're going over 3 gallons a minute, depending on how dirty the influent is, you can quickly overwhelm those sieves. So having flow control over the volume is definitely necessary. We chose stainless steel transfer tubing for everything. We didn't want nylon, PTFE, or any type of materials that could possibly add contaminants to our sample and be miscounted. Programmable timer. Now, at 3 gallons per minute in one of our influent samples, will probably bind up and clog that screen mesh in about 3 hours. And so I would typically set my timer for 2.5-hour intervals. We'd go out, collect the material from the sieve, reinitiate the pump for another 2.5 hours, and we would do that over a 24-hour period. So we're getting a composite sample over a 24-hour period. Sieve stacks you can get in various porosity diameters, and then some type of a collection sieve. Here it's showing a plastic bucket, but we took a regular aluminum bucket, drilled holes in the bottom that contained all the sieves in it. One other key feature that you should consider is an inline flow meter. Inline flow meter will help you dial in the flow control of the volume going to the sieves. That'll avoid overburdening them, but also a flow controller that also is a totalizer and records total volume processed. That's very handy. And so the ideal features would be something that's easy to set up and calibrate, something that allows flow control, something that records total volume. You can use peristaltic or submersible pumps, and fractionating sieves. So the photograph on the left is of a typical influent sample. You'll see large debris, a large mat that's largely toilet paper that's binding on the filter, while the one on the right, that's a much finer mesh. That's probably about a 25 micron sieve, and it's free of debris, still has some cellulose material, but a lot of algal material. A lot of our tanks are open, exposed to daylight, so we get a lot of algal buildup in there, and that's what you see on the sieve itself. So the real challenge comes in the sample preparation. And again, the SOP that we're trying to develop needs to be easy to replicate, needs to address interferences, and minimize the loss of particles. One item not on here is minimize the addition of particles. If you have an aggressive sample preparation scheme, and it's overly aggressive, even just stirring the sample overly aggressive, one particle could break into 2, 3, multiple particles, and then numerically you're overcounting those particles. So we want to keep that in mind as well. So one of the test methods I didn't highlight on the 13 methods that we evaluated was this one by Loader in 2017. It's the Universal Enzymatic Purification Protocol. This is a very elegant protocol because it addresses a variety of interference that one may encounter, especially in wastewater matrices. It addresses proteins, fats, oils and greases, toilet paper, cellulose, has a wet peroxide oxidation, chitinase, that is a lot of the, you're going to see bug fragments that are in sewage as well. So we chose it based on these 3 key characteristics, and in a slightly different order. Almost all the methodologies we encountered use some form of wet oxidation with peroxide, and a few of them used density separations using either zinc chloride or a calcium chloride. And when dealing with wastewater, cellulose is a huge burden. It's probably 90-95% of the problem that you're trying to address is the interference from cellulose. And so these 3 steps we took, and we chose to improve upon them, because looking at here, look at the amount of time it was required to do this analysis. It ranged from well over a week up to 15 days to prepare all these steps. Keep in mind, each one of these steps you're adding something to the sample. It's exposed to your lab environment. So if contamination is ubiquitous, if it's everywhere, you're going to contaminate that sample at every step along the way. And so we chose these 3 steps outlined in red, but going away from the enzymatic protocol. We needed faster turnaround time and to avoid contamination to make this a viable test for most laboratories. And we also chose to implement some type of protocol to validate our study. So we had method spikes included as well. So this was the samples that we analyzed using the Loader protocol with the enzymatic buffers. And you'll see there is some cellulosic debris still present in the sample, but it's probably 99% removed from what it was in the original wastewater sample. You see these large particles. These were obviously an influent sample, and we spiked them with 2 different types of polymers. And I've highlighted those here. The red polyethylene polymers these were 70 to 120 microns, and the larger particles were another polypropylene that was in the 200 micron range in size. And so we spiked with two different type of polymers, and when you visually look at them under a microscope like this, if the particles are broken, then you can it leads to overestimation, but it tells you that your preparation technique is probably too aggressive. And if you don't recover your particles, you're probably assured that you're losing material throughout your process as well. And so this was very valuable for us. Our final method selection uses those three key features that we found common to most analytical methods, but in a slightly different order. We chose as our first stage to do the wet oxidation. Almost 90% of the research articles use some form of wet oxidation, and we find that extremely valuable because, for one, it oxidizes a lot of the organic matter and the easily oxidizable carbon, and so it's a form of disinfecting the sample and protecting the laboratory staff before moving forward. We do this with a Fenton reagent, which is a 30% peroxide with an iron catalyst. This will destroy pathogens. It oxidizes the organic matter in the sample, rendering it much cleaner, especially in those effluent samples where you have a lot of algal material. And it also releases materials from the plastic surface. So one of the things that EPA was concerned about was the amount of biofilm and co-contaminants that may be deposited on a sample particle, plastic particle. And so when you think of that, we commonly see PCBs and other type of materials that are bound to plastics, and if these are in an ocean or in a lake or a waterway, the aquatic species will then come along, eat those plastics, ingest those co-contaminants as well. And so that's why it's critical for us to know the shape and size of the particle to estimate how much co-contaminants are on there as well. And the Fenton reagent releases that biofilm and those contaminants from the plastic surface. The next step was the use of a Schweizer's reagent in place of the cellulase digestion. In the loader method, they used a cellulase digestion, which proved quite effective, but it was very timely, often requiring up to 3 days to remove the cellulose toilet paper from the influent samples. And so a Schweizer's reagent is actually a copper ammonia complex, and it's almost like something a magician would use. You add the Schweizer's reagent to the sample, it instantly breaks down the cellulose and allows us to then take that same sample, run it through the corresponding sieve, and the cellulose is gone. We then extract those particles right off the sieve a second time. It's a rapid reaction. It eliminates cellulose. It greatly minimizes sample preparation time, and it avoids contamination. The third key step was density separations. Throughout the literature, we'll see a lot of laboratories using either a zinc chloride or a calcium chloride solution at a concentration that makes it to a density of 1.4 to 1.7 grams per mil. In effect, the heavier inorganic type material sinks to the bottom, the plastics float to the plastic polymers float to the top, then those are then skimmed off and analyzed. One problem that we note with that is you'll see a lot of those plastic particles that float to the top want to adhere to the edge of the container, which can be problematic. And so for our studies, we chose to use a centrifuge rather than floating the material to the top. And I'll describe that briefly for you. So here's the way the procedure would typically go. And depending on the sample matrices you're using, all of these steps may not be necessary. So this is a worst case scenario using wastewater influent. We'll do a wet peroxide with a Fenton reagent for an hour. That's going to decontaminate the sample and remove all the easily oxidizable carbon and lift any biofilm off of the plastic particles themselves, rendering them a much cleaner surface for spectroscopy analysis. We would then centrifuge that sample for 15 minutes. All the plastic particles are going to become compacted in the pellet, and we decant off the excess liquid using a micropipette. Then if you were using wastewater samples, we'd do a cellulose digestion with a Schweizer's reagent for 5 minutes. Again, centrifuge and decant, and then quench that with ammonium hydroxide 5 minutes. This is in comparison to the multiple day digestion that we saw with the loader using the catalase enzymes. The next step is we use a tris buffer, and that tris buffer, what that does, it helps keep the particles in solution and from adhering to the plastic walls of the container itself. And then we use if you are still using wastewater samples, you may want to do a simultaneous protease and lipase digestion. Obviously, for effluent samples, surface water samples, or bottled water samples, you're not going to have proteins and fats in the sample, and so you can avoid that step, which is, it's a 20-hour step, but that's because it has these enzymes that need time to react. And again, you would then centrifuge, decant, wash it, and be ready for analysis. We always suspend the plastic particles finally in methanol, and that is to keep it from adhering to the walls of the container, keeps them in solution. And then when you plate the particles out onto a microscope slide, you're able to, the methanol will evaporate, and so now you have a clean matrix that you can put in the instrument. So essentially, it's a two-step process for most samples. One, using the Fenton reagent to decontaminate the sample and clean it up, and then the second, using a Schweizer's reagent for removing cellulose. Now again, you only need the Schweizer's if you're dealing with influent samples that have a lot of cellulose material in there, a lot of toilet paper. And we typically do these, although they're pictured in beakers here on the screen, we typically do all of this within a single conical tube, and that same tube is used throughout. Therefore, there's no transferring of solutions from one beaker to another where you could possibly encounter contamination. Everything's done in a single vial. So you'd add your reagent to the vial, shake it for the time interval, centrifuge it, open the vial, put in your second reagent, shake it, centrifuge it. So we're trying to minimize contamination throughout this process. So this is an oversimplified cartoon showing a micro centrifuge tube, but it would be the same for the larger 50 ml tubes that we used. And so after centrifuging the final sample, all the material is compacted in the pellet because we didn't use any density separations. And then we use a micro pipette to basically pull off all the liquid from the sample, getting as close to the pellet as you can. And you can do this under a stereo microscope as well. So you make sure you're not pulling up any of the particles in the pipette. And so once we've pulled out all that material, we add just a drop or two of methanol. We then remove the pellet, plate the pellet out on the microscope slide. This eliminates filtration, sample loss. The cellulose is completely removed. Preparation time is very short. We can do this in just a few hours, taking a sample from the initial raw sample collected off the sieves to a sample that's ready for analysis. All the sieve contents are now plated on a single slide, and we have reference spike recoveries that are near 100%. And finally, we get to the sample analysis workflow. So regardless of what method we're using, whether it's micro Raman, micro FTIR, or GC pyrolysis, we want to be able to positively identify the particles, and the way we do that is against a reference library. Quantification of particles, you're not going to be able to quantify the number of particles using GC pyrolysis. You will get a total mass of the particle content, but if you're using micro FTIR or micro Raman, you will be able to count the individual particles. They will also allow a particle size distribution, which is going to be a beneficial feature. And provided you have a focal plane array or some instrument that's able to rapidly scan the slide, the results should be fast and reproducible. So after working with micro Raman method for a good part of a year, we were able to obtain Agilent LDR 8700 as a chemical imaging system that was originally designed for pharmaceutical analysis. However, it has demonstrated itself to be very beneficial for particle analysis as well. So the first step is to take that slide that we previously plated, we insert it into the instrument, and the first thing we select is the relative sensitivity. We would pull up a library. The microplastics library that we've assembled has common components such as polyethylene, polypropylene, LDPE, HDPE, Teflon, and other common rubbers and different types of plastic polymers. We then select the sensitivity. For this slide, this is influent sample, so I've only selected to analyze for particles larger than 100 micron and smaller than 5,000 micron. Now, if we were looking at effluent samples, we may change this to look at particles from 20 micron up to maybe 300 microns. This was material obtained on a 500 micron sieve, and this area that's under analysis, this probably took it roughly an hour to analyze, and it's probably about 1/6 of the slide. And you'll see the number of particles that were analyzed and identified on this. Here's the most important part. Every particle that we've analyzed, you get an optical image, and then you get the infrared image, along with the spectrum and the reference spectrum overlaid. And you also get the benefit of the diameter and area of the particle. And this is something that EPA was very interested in for establishing what the co-contaminant loading may be on these particles. And so these were a polystyrene particle. Here you see the data. For that entire slide, you see the number count and the number of particles that were detected, what percentage that they represent of the total particles identified. And for any particles that are unidentified, that are unknown, you can always go back and manually analyze them with an FTIR and maybe look at against different libraries. Now, this is just the natural microplastics library, which you typically see here. We still have cellulose on screen because we know we're dealing with the wastewater matrices, so we look for that. But here's some normal sample analysis, and it tells you what the particle size range was for each of those particles. So for this wastewater influent, we'll see that almost 95% of all of our particles are in the 20 to 50 micron range, with the second tier being in the 50 to 100 micron range. Very valuable information we're able to acquire. So thank you for your time today. Here's my contact information if any of you would like to get in touch with me. If you have any suggestions for improvements, please feel free to reach out to me. Thank you.